Hello all, welcome back to the BioRocks channel. Today in this video, I'll be talking about the asexual reproduction methods employed by the plants. Yes, so today's topic is all about asexual reproductive techniques employed by plants. So you might be learning in your 11th standard regarding all the different sorts of reproductive techniques. That is, they are going to reproduce through structures called as spores. But you might be finding some different types of spores and how to actually identify and what are the differences between these. Today I have come up with this video to make it very short, sweet, easy for you all bright students. Number one, the type of spores you can see are zoospores. You might be coming across aplanospores. You might be coming across chlamydospores. You might be coming across some other sorts of spores, which may be the zygospores. And you might be coming across some other different techniques of reproductive asexual methods by algae and fungi, like they might be akinetes. Okay, we will be going through these. We'll just get into the details and find out that what are all these sorts of things. But just remember, children, that zoo is something dealing with animals. So definitely these spores are motile spores. If they are motile, then they have to possess some locomotory things. Then we have, if something is non-motile, then we call it as aplanospores. Aplanospores. If they are very, very, very thick-walled, we call them as chlamydospores. Besides these, we even have some other spores which are usually employed by the parasitic fungi, which are being called as conidia. Okay, these all are the ones that you can see. And remember that zoospores can otherwise be called as planospores, which are motile, which can swim. Okay, and if the plano, the movement is not there, we call it as aplanospores. Okay, usually the planospores, which are motile, will be present in the waters, whereas aplanospores, which are non-motile, will be usually produced on the land during the adverse conditions on the land. Okay, these are a few things. Remember, these all, I'll be coming across every one of it. So first what we'll do is, we will get into the details of all these spores. So first I'll be touching up here Thallophyta, that is here Thallophyta. What is Thallophyta? Thallophyta is nothing but, this is the division that comes under the cryptogams. Okay. I think you all know what are cryptogams. So what are cryptogams? Cryptogams are those which are the flowerless, seedless, spore-producing plants. Okay. These are flowerless, seedless, spore-producing plants. And under the cryptogamy, we have Thallophyta, then we have the Bryophyta, and then we have a Pteridophyta. I hope you all have once gone through the classification and you have learned in your 10th standards about the classification. So I'm talking about the same, that your cryptogamy is comprising of some divisions like Thallophyta and Bryophyta, and pteridophyta. Okay, I'm coming across thallophyta to explain these all spores. Okay, and the example that I'm taking here for the thallophyta is, first I'm talking about the algae. We will be learning about algae as well as we'll be learning about the fungi under the division of thallophyta. And why do we call it as thallophyta? Because their vegetative body is called as thallus. So we are calling them as thallophyta. So when it comes to the reproduction technique in asexual reproduction, usually, now the example that I'm talking in the algae is about the Spirogyra. So how it reproduces asexually is, there are some techniques by it. Either it can be producing the non-motile spores. So these are all the asexual reproductive techniques employed by Spirogyra, which is an example of the algae. Okay, so it's reproducing by asexually by producing non-motile spores. Okay, and these non-motile spores are akinets or the aplanospores. Akinets or aplanospores. It might be employing any one of the technique. But what is an akinet? Akinet is nothing but, for example, if I'm drawing a Spirogyra, and if these are the cells of the Spirogyra, what happens is, in akinets, this vegetative cell which is being present, this vegetative cell is going to develop, or it is going to produce a very, or it is going to actually form a very thickened cell wall here. Okay, it's going to produce a thickened wall around the cell. It's going to form a thickened wall around the cell. So akinet, what it is doing, it is formed because of what? Due to the development of a very thickened wall. Or what happens is, the cell itself is going to have the primary wall that itself is going to be thickened more. Why? Because it has to suit and cope up the unfavorable conditions. Okay, so it has to cope up with the unfavorable conditions. So the wall is being thickened a lot, and now it is forming a structure, and this structure will be called as akinet. Akinet. If at all, if there is return of the favorable conditions, then what happens? Then your akinet which is being present, this is going to germinate. This is going to germinate, and this will give rise to a new filament again. This is the way it's trying to actually protect itself from the unfavorable conditions. Got? The next one that we are having is aplanospores. And what is an aplanospore is nothing but, in few of the spirogyral species like Spirogyra aplanospora, here what they do is, they are multiplying by this technique which is being called as aplanospores. Now, what is aplanospore is nothing but, for example, if this is my Spirogyra and these are the cells, and if at all, if this is the protoplasm filled inside, now the cell, what it does is, the protoplasm is going to contract like this, and it is going to round up, and it is going to develop a new wall of its own. So now it is developing a new wall of its own. Okay, and like this, we can see that this structure which is being formed is an aplanospore. So what has happened here is nothing but, the protoplasm of the cell is contracting, it is rounding up and developing a new wall of its own, and that is an aplanospore. And the aplanospore, what happens is, this is going to get released. When is that? When this parental Spirogyra is going to decay, when it's going to decay, then what happens is, your parental cell is going to release this aplanospore out. And now this aplanospore is going to germinate and produce again a new filament. This is how they are going to reproduce. There is no involvement of the sexes, so we are calling it as purely asexual reproduction. Is this clear now? Now, next coming on to another, which is a technique that we are going to call of production of the zygospore. You might have learned in your 11th that the algae, usually the spirogyras, they are, even in paramecium, conjugation, you learned about conjugation, right? So what we see in conjugation is, this is actually a way of a sexual process, okay? This is regarded as a sexual process, okay? The reason is, here we see that there are two cells, or we usually call them as gametangia, which are going to involve and they are going to unite. And that union of this gametangia or cells is called as conjugation. Okay. And these two cells can be of the same filament of the spirogyra, or it might be between the two cells belonging to the two different filaments of the spirogyra. It might be between these two, the exchange, or it might be between the two cells of a same filament. It can happen like this. Okay. Now coming on to, if the two cells are belonging to two different filaments like this, we call this conjugation as scalary form conjugation. Okay. And if the two conjugate cells are belonging to the same filament, then what do we call this as? We call this as lateral conjugation. Lateral conjugation. Now, I'm coming to the process of formation of the zygospore. Now, you need to understand this one. So, remember that the two filaments that are coming and lying on opposite and are parallel to each other, right here, that you can see in this procedure. So, now what happens is the filaments which are being present here, one sec, this is going to be the ones, yeah. So one of the cells will be acting as a male and the other acts as female, and the male filament, what it does is, it is going to produce, first of all, a small protuberance like this, an outgrowth like this, that you can call it as a papillae. It produces a papillae. Towards which side? Towards the female filament. Okay. And now what happens is, this is the first thing that the male has produced of papillae. Now the second stage, what happens is, the female filament, the cell of the female filament is going to produce, or it is going to be activated. And this is going to be either stimulated, I can use a term as it's being stimulated. And now this is also going to produce its own sort of a papillae this way. Now this has been stimulated, so it is also producing a papillae. Now what happens is, both the papillae of the opposite cells are going to fuse. So that is going to be in this manner. So both of them are going to fuse. So even the female papillae like this, they have been fused. And they are going to produce a tubular conjugation process like this. And here the wall present here will be dissolved. It will be dissolved by the action of an enzyme called as synthase enzyme. Now what happens? Check out. Now what happens is, you can see that both of these papillae have fused together, and now there is, you can easily see that the things can move in, and this tube which is formed now is being called as a conjugation tube. This is called as a conjugation tube, okay, between these two opposite cells. And now remember that as most of the cells of the filaments participate at a time, it's not that only one cell will be transformed and only the other here, but in one single filament, there might be multiple cells acting as males and the other multiple cells acting as females, and the process will be taking up. Okay, this can happen. So it looks like as if you have built up a ladder between the two cells, isn't it? So we are going to call this as a scalary form conjugation. Now what happens is, for instance, if this is a cell here, now what happens, the protoplasm of the male cell which has been present is contracted. Just a while ago, you have learned that a contracted protoplast is an aplanospore. So isn't it that the male cell is also trying to produce an gamete same as an aplanospore, but I call this as what? We call it as a aplanogamete. And this is a male one. And this is not going to be perfectly like this. It may be amoeboid shape, but the protoplasm has contracted. Now what happens? The protoplasm of the female cell contracts only to the extent of accommodating the male gamete. Okay, so it's going to be like the female gamete protoplasm is not going to, wait a minute, is not going to contract massively like the male, but it sees that it is going to actually accommodate, it can create that space for the male to get inside, and this is your male aplanogamete that will be coming up here. So that is the only space created by the female, but the contraction is not very severe as in the male. So males are having complete protoplast fusion, there is contraction, but in females it is not the work case. She's going to only create a space to create the place to accommodate your male aplanogamete. That's it. Now what happens is the male gamete will move through the conjugation tube, it has reached already the female. Now they both are going to fuse together, and haploid and haploid, this is going to result in the formation of a diploid zygote. Okay, so diploid gamete, there is a zygote is being formed here, and now you can see that this is going to, your zygote is going to secrete a thick wall. Let me create a thick wall around this now. So the zygote has created a very thick wall around it. And now this is going to be the resting zygote and a resting spore. This is a resting spore, and this spore is none other than your zygospore. Have you understood what is a zygospore now? I hope I made it clear for you all. Okay. So now you learned about the zygospore. And what have you learned? You have learned about what are echinates. You have learned about the aplanospores. Let us see. We have learned about aplanospores, right? We have learned about the zygospores. We have learned about the echinates. Then we are left over with what is meant by a zoospore now. Okay, zoospore. What is a zoospore? Zoo is nothing but what I told you, planospores, which are motile spores. So how are they motile? They are going to be motile because these particular spores, the motile spores, are going to have the flagella like this. And these flagella will be making the movements, whipping movements in the water, due to which these spores can actually be motile. Then we call these spores as zoospores. Okay, is this clear now? Those are the zoospores that are being present, and even fungi. Now we have seen the examples of the, what are those that we have seen just now? We have learned in Spirogyra, that is algae, we have learned about the echinate, then we learned about the aplanospore, then we learned about the zygospore. But remember that in fungi also, we can see asexual reproduction. And in asexual reproduction of your fungi, which is also one of the classifications in your, which one is that? Thallophyta. So even algae and fungi are belong to Thallophyta. So fungi is asexually reproducing again by aplanospores. Aplanospores. But these aplanospores are usually called as sporangiospores. Sporangiospores. Okay. And sporangiospores usually will be produced mostly during the favorable conditions only. When there is good amount of food, good amount of supply of the environment conditions and all. But during the unfavorable conditions, the aplanospores that it produces are being called as chlamydospores. Chlamydospores. Now, this is important for you to remember. Okay. Then coming on to asexual reproduction, as I told you that during the unfavorable conditions, especially during very, very, very dry conditions, what happens is the protoplasm of the hyphae. First of all, let me draw. This is going to be the rhizopus that I'm drawing up. This is your rhizopus, and we are having some rhizoids like this. Okay, these are the rhizoids. Now what happens is in the hyphae itself, you can see that some of the protoplasmic contents. So let us draw some hyphae like this going up. So these are the branched hyphae here, branched hyphae. So the hyphae protoplasm which is being present here is going to accumulate. The whole protoplasm is coming up and it is accumulating in one area. Okay, and the other areas are becoming empty. The other hyphae areas are becoming empty. Only one area, all the protoplasm has gotten accumulated. And these localized accumulation secretes what? A thick wall around it now. So let us draw a thick wall around that. So it is developing a thick wall around itself. And this thick wall, which is being created here around the protoplasm, is none other than your chlamydospore. Chlamydospore. So this is nothing but what? Aplanospore, right? Let us compare the aplanospore now. Where is aplanospore? This one, the protoplasmic contraction. Same thing is happening here, and it has formed the aplanospore that is being called as chlamydospore in fungi. That is, we are taking the example of rhizopus, which is a bread mold. Bread mold. So in rhizopus, you can see even the formation of this one. But remember, they survive during the adverse conditions. These chlamydospores will be surviving during the unfavorable conditions. But once your favorable conditions return, these are going to surely germinate and they will be producing the new mycelium. What is meant by mycelium? The vegetative body of fungi is being called as mycelium. So again, they are producing some new filaments in the algae, and here they're producing new mycelium, new rhizopus here. That is about the asexual reproduction process in these plants that you will be coming across during your syllabi. Okay, we have learned about the zoospores, aplanospores, and the chlamydospores in fungi and akinets. Now, let me show you all the different types of the spores that we have learned today. Okay, so, yeah. So, the one that you are seeing on the screen is a spirogyra, where you can see that there are the conjugation tubes that are being formed here, and they are trying to exchange. You can see that the female has already been creating up these spaces, and there is movement of your, let me take this, 1 second. You can see that the females have created space, so the male aplanogametes have moved in. There will be formation of surely a zygospore here. That is in your spirogyra. Now, next coming on to another that I would like to show you is the, wait a second. Now, the image that you're seeing on the screen is about the aplanospores. As you can see in the spirogyra, there are few cells where the protoplasm has contracted. They have formed some new walls around it. And this one that you're seeing here typically are nothing but your aplanospores. Aplanospores. Okay. Now, we'll be watching another, and that is going to be your zoospores. So the one that you're seeing on the screen now is a zoospore, which are surely having the flagella. Okay, so let me come to this. So the spores, which are going to be motile, which have their own flagellae, which can enable them to move in the water currents, is going to be the zoospores that you are now seeing on your screen. Okay, and the next one that we'll see is about, a moment please, and that is going to be your asexual spores, which are the thick-walled spores again. What are those thick-walled spores that we are going to have? Once again, the aplanospores. These are also the aplanospores, where you can see that they have actually formed this coloration, thick-walled structure around the protoplasm, which has been present, that you can see here. That is the one which is an aplanospore once again. Now, coming on to the next one, we have seen, yeah, this is your fungal spore that we are having. These are the zoospores that we have seen with the flagella. Then we have seen the aplanospores here in the Spirogyra. And the next one that we are having is these akinites. What are the akinites? The resting spores. I told you that akinete is a one which is going to actually, give me a second. Yeah, akinites are those where you can see that the vegetative cell itself is going to secrete some, what you call is a thick wall around it. And once the complete parental filaments are going to decay, then this akinete is going to germinate into a new individual. And those thick cells that you can see on the screen here are, these ones are the ones which are going to be the akinites here. Okay, these are the different ones that I wanted you to know about in this video. Now, let me share the things that we have learned. So we have today learned about the asexual reproductive techniques that you will be coming across all through the botany of your 11th standard. So you need to know the basic fundamentals of all those things before you jump into your NEET. I hope this video is helping you in understanding this one. Thank you all. And if you have loved this video, please do like, share, and subscribe my channel. Thank you. Thank you all. Thank you.